In this last section for the video part of the lecture, I want to talk a little bit about how when you have multiple gene pairs that have multiple types of inheritance, so you're doing a dihybrid cross basically, but they have different patterns of inheritance, not that classic dominant recessive relationship, you start to get phenotypes that are outside of that 9:3 to 3:1 ratio. So an example would be if you consider perhaps the combination of albinism and blood type. So albinism is inherited in a classic dominant recessive relationship, but we also know that blood type is inherited via multiple alleles where you have a codominance relationship between A and B, and then a dominant relationship between A and O and B and O. So the way we would look at that, if both individuals were heterozygous, so they're both carriers for albinism, we would represent the dominant phenotype as A, the recessive as little a, and then we have our type AB blood, which involves both alleles. So we can do one of those kind of separate Punnett squares, where we look at the albinism first. And when you cross two heterozygotes, because this is a classic Mendelian type of genetics, you get that 3:1 ratio. When we look at the blood type, again, they're heterozygous, but because A and B are codominant to one another, the heterozygote will express both phenotypes. So you have type A, type B, and then that type AB. So in order to calculate the probability of the phenotypes, and by probability of the phenotypes, I mean pigmented with what blood type, albino with what blood type, you do one of these charts like we've done before. So when you cross the two heterozygotes and you're only considering the gene responsible for albinism, you end up with 3/4 of your offspring that have normal pigmentation and 1/4 that are albino. Then, of all offspring, you have to consider the blood type. 1/4 will have type A, 2/4 will have type AB, and 1/4 will have type B, and you list them for each of the pigmentation options. And then you multiply across. So even though we're doing a dihybrid cross with two heterozygotes, instead of that 9:3 to 3:1 ratio that we're familiar with, right, we would normally expect 9, 3, 3, 1. We're actually getting 6 different phenotypes. So instead of 4 phenotypes, we get 6, and they are in a completely different ratio. Now, I don't expect you to, off the top of your head, remember what the ratio is, because when you have extensions of Mendelian genetics, the ratios can be different depending on the inheritance patterns. But I do want you to be able to generate an image or, the equations, something like this. Now, we've talked about this idea very briefly when we talked about that Bombay phenotype, that you can have multiple genes affecting a single phenotype. And that can also make it a little bit more difficult to predict what's happening in terms of inheritance patterns. And so you have to consider these gene interactions when you're talking about developing a phenotype. So in a gene interaction, you have several genes that are influencing a characteristic, right, the phenotype. And the reason for this is that with each of these genes making a different product, all of those products are important for making the phenotype. And so one of the terms that we use when we're talking about multiple genes influencing one phenotype is epistasis. So epistasis is when you have two pairs of genes that are influencing a phenotype. And the expression or the effect of one gene can modify the effect of the other. And this definitely could be something like that Bombay phenotype. So remember when we talked about the Bombay phenotype, we have these three, blood type alleles, the IA, the IB. I call it O because I think that's a little bit less complicated, but we can actually call it the real name, the real allele symbol, and we can call it I, right? So you have these three combinations. But then we also talked about there was this other gene called FUT1. And there was this woman who looked like, right, the phenotype was that she was type O. But genetically, she actually had to be a carrier for type B. And it turned out she had a mutation here which affected the blood type. So that's a type of epistatic interaction. The blood type genes were normal, but that other gene, that FUT1, affected how the phenotype actually showed up. And as you start to look at more complex organisms with organ systems and, multiple organ systems interacting, you really do start to see that the complexity means that there are going to be all of these different genes interacting to affect single phenotypes. Another example, I gave you that blood type Bombay example. Another example is in hereditary deafness. So, getting an ear isn't just like, there's not just a gene that's like, this is what your ear's going to look like. There are lots of genes involved in shaping what your ear looks like and how it forms. And so when you have a situation where one of those genes, or maybe some of those genes, have a mutation, you can end up with hereditary deafness. Basically, the person is born either hearing impaired or with complete hearing loss. And so they call this situation like heterogeneous. There are lots of different genes involved in making the ear, both the internal and external structures. And so when you have an issue with one or more of them, you can have an issue with the ability for the ear to function properly. And again, here's that example of the Bombay phenotype I told you about before, where you have the mutant gene, the FUT1, when it is mutated, prevents the expression of the blood type encoded by the genetics. So this is what that would look like here if you had individuals that had type AB blood that were heterozygous for the Bombay phenotype. You could do the consideration of the blood types as one cross, and then you could look at the cross of the H substance, the FUT1 gene. And when you do that, you get your same blood type patterns that we talked about before. If you have two individuals that are blood type AB, they would have offspring that are type A, type AB, and type B. So those are listed here. But then you have to consider this Bombay phenotype. So 3/4 of the offspring should have the normal H substance. 1/4 would have the mutation. So if you list those out here, like is done in this image, you get these probabilities. Now, when you're looking at the phenotype, so this is a type A blood, but this person does not form the A substance. So you would think they should be type A, but it turns out phenotype is O. We see the same thing here, AB, but do not form the H. So they should be AB. But because they don't form the H, even though genetically they should be type AB, they're actually O. And we have that final example here where the individual is type B, but does not form the substance. So even though they should be type B, they are in fact type O. And so that's two examples in humans of epistasis. So gene interaction can reveal these different types of inheritance patterns. And when we think about, like, what can happen here, there are effects on various phenotypic categories. And so we're going to talk about how epistasis could maybe be like recessive or dominant or incomplete or codominant. And so they give this example here where if you're doing, again, a classic dihybrid cross with Mendelian genetics, these are your phenotype ratios, the 9:3 to 3:1. And the phenotype ratios, right, 9 of the organisms would look dominant for both. 3 of the organisms would look dominant for one and recessive for the other. 3 of the organisms would be opposite, recessive for the first and dominant for the second. And 1 would be recessive for everything. But depending on each of the 4 epistatic types of interactions, right, you can start to see multiple different types of phenotypes result, or phenotypic ratios. Okay? And again, these are not situations where necessarily you're going to remember the phenotype ratios. 9:3 to 3:1 is pretty easy, but you should be able to set up some of these problems and calculate what those different ratios might be. So with recessive epistasis, that's that same idea or that same concept of the Bombay phenotype. Even though blood type genes are normal, when that other gene is recessive, it affects the way the individual does the blood type. And so the example that they're giving here is, again, in mice with their agouti phenotype. And so if you are looking at, this is not the same as the agouti in yellow. This is not that mutant yellow. This is the agouti dominant and the irregular recessive. And so we'll look at this on my screen. We'll draw this out. Okay. Okay. All right, so just to refresh you one more time, what we're looking at, right? If an organism has, just looking at coat color, right? If an organism has 2 big A's, they're agouti. If they have 2 little A's, they are black. Okay, so let's look at our Punnett square. So we have big A, that mouse should be agouti. We have big A, that mouse should also be agouti. And then the allele, the little a allele, if they're recessive for that, the mouse will be black. But then on this other slide, we saw, they talked about this gene called B. And so B also has a classic Mendelian inheritance pattern. And so big B is normal color. So B actually controls the deposition of the pigment in the hairs, where A controls the color, B controls depositing the color. So as long as a mouse has 1 dominant copy of B, they are able to deposit their color and they will be normal, whatever that normal color is. If a mouse is albino, or if a mouse is recessive for B, the mouse will be albino. It can't deposit color. So we know that if we do a cross of 2 heterozygotes for coat color, we get that 3:1 ratio. So 3/4 will be that agouti color, and 1/4 should be black. But now we have these options here too. So in this case, all right, with looking at crossing for gene B, we get 3/4 normal color, 1/4 a lack of color or albino. And the same thing over here. Okay, and then we just do our multiplication, 3/4 times 3/4, 9/16. We'll have normal agouti color. 3/4 times 1/4 will be albino, right? Because the lack of color means no color. Okay, if we go down here to the bottom, 3/16 will be black. And then 1/16 again, albino. So actually, when I now start to combine these ratios, it looks at first like I have 9:3 to 3:1, but look, these two are both albino, so I have to combine them. So instead, what I really have is 9/16 agouti, 4/16 albino, and 3/16 black. So this is a type of recessive epistasis, but you see it now gives me a different ratio. And of course, again, I don't expect you to remember the ratios off the top of your head, but to be able to do a problem like this will be really important. Okay. And again, depending on how the phenotypes work, these are various conditions in different animals and plants, the characteristics that you're looking at, and the different ratios you get. So if the relationship is described to you, you should be able to do one of these types of problems.